I think we're ready to go. We're good? None? Good. If you don't mind, I'll say a few words.
Okay, go ahead, yeah.
Okay. So it's a great pleasure today.
What?
You have control of the speaker. That's a hint. And I'm sure to explain to you, but just so you know, we have an internal audience and a larger audience. So,
Yeah, no, try to.
So Dr. Evavold is at the University of he's here. He's the George Weber of Immunology, and has really sort of a focused history, which I adore. That to be quite impressive with being a serious scientist. Got his bachelor's degree, right? And a PhD at University of Chicago on T-cell responses, and then at Tennessee, that continued postdoc at Washington University, again on T-cell responses and responsible peptides, and then the experimental practice at Emory, right afterwards, up through mid-career at Emory, and then came here in 2017 as a professor in the Department of Microbiology and Immunology, and a division chief of Microbiology and Immunology. And in 2019, also became the vice chair of research positions today. Maintaining incredible NIH funding, publications, including second senior at this point. There's, I think I'm looking at how quickly the room filled up, preaching to the choir here, but it turns out immunology is important. And it's important to every organ system in the body, and even cardiovascular investigators now realize immunology, how important immunology is. And so we're learning a lot. You know, I'm sure you're going to throw out statistics like this, but I'll say just in case. At least half the heart, any of our hearts, do not consist of cardiomyocytes. Okay, keep that in mind, and keep in mind what they regulate. Cardiomyocytes in homeostasis, the regulation of inflammatory response after injury, both acutely and chronically, and certainly during things like infection. So this has become a fascinating new area, and all of us are obliged, frankly, to learn more. Unfortunately, we have one of the nation's experts in T-cell immunology here at the University of Utah. I want to just also add how much I've enjoyed as I joined the campus community here. And so, for multiple reasons, this is special. Girl, welcome.
I'm saying that you'd have something. We're doing EEJs. I do take lots. I've been coming. Basic science working group abbreviation is BSWG, and you know what BS means. So we're this university is kind of unusual in that we don't have a department of microbiology and immunology, so we are a division. And so I was recruited to be the division chief about 5 years ago. And so we've expanded our huge roles, microbiology and immunology, especially on the immunology side. So if you have questions on immunology, this should be. So, yeah, we can get started. So, and also, please feel free to ask questions whenever. Even immunologists with a lot of what I talk about is new concepts. 15 years ago, I started working with fundamental engineers at universities in Atlanta, Georgia. And what I've been trying to do, as Robin pointed out, I'm trying to figure out what's his whole life, even as no one will really keep trying. And what we did was kind of learn about about a dozen years ago, a little more than that, went out to a seminar with an engineer, and never heard of. I know concepts of affinity, how things run, but I never heard that when we do a chemical measurement for affinity, that's in 3 dimensions because it's in solution. And T cells, or almost every cell of the immune system, they interact in 2 dimensions. And those parameters of affinity and bond lifetime are very different. That's what I started doing 7 years ago, and ever since then, it's really solving T cells. So the title is during immune response, force awaken T cells, and that is kind of lifesaver. So we are doing that thing. Currently, I have no conflict of interest in this. And if we started, just with a primer of what an immune response looks like, T cells are important parts of the immune system. There's millions of T cells in the body. Each T cell has a unique T cell receptor, critical for all adaptive immunity. So they regulate all cells. They provide cytokines to get macrophages to work better, neutrophils to work better, B cells to work better. Autoimmune disease is always caused and release initiated by T cells. Anti-tumor responses, all the immunotherapy right now pretty much target T cells. That's what you're trying to improve. Of course, vaccination and infection. This is a picture you could take out of almost any immune book. But this is a ribbon diagram. This is the T cell receptor. And T cell receptor interacts with a protein called MHC, human HLA. You may have heard of those abbreviations, and that protein, MHC, is built to present peptide fragments to T cells. And that peptide fragment is what triggers the T cell. And the immune response macrophages, dendritic cells, B cells, are always chopping up proteins and peptides from COVID, peptides from flu, peptides from cell. And T cells are going around sampling those MHC molecules to say, should I trigger, should I get activation? And so really, this interface here between the T cell receptor and the peptide MHC is what I've really been focusing on to see how it regulates response. And specifically, as I said, about a dozen years ago, we got into trying to figure out affinity on lifetime. That's really how you decide how a receptor can trigger. And then up here, I just put this picture of, not my work, but a scanning EM of a T cell. I like it because it shows all the projections a T cell has. It's not a very flat surface. You have to remember that interacting surface. Also, what I like about this picture, it got published twice. Originally published here, and then they colorized it. Change colors. Anyway, so overarching questions for the adaptive immune responses is fundamentally what does the immune system do? You can say it in a lot of different ways. They would have to respond to everything that's possible in a peptide sequence. So it's 20 amino acids. That's a lot of possibilities. And then you add the 3-dimensional surfaces. And so, but your immune system will respond to it in some shape. And so that's always been a driving question. How does that work? And then a big question from that is, how do you distinguish yourself from non-self? So you may think you're different than the virus or bacteria. And we have protein sequences that look very much like self. And so the immune system is always fighting to figure out, should you be attacking this? Is this an infection, or is this your immune myelin sheath? And you don't want to attack. And then are there, what I'm doing now, are there kinetic factors that determine success? Then basically, why are we interested in kinetics? So basically, affinity and bond lifetime dictate response. And for T cells, it basically determines fate. So they have a huge ability to respond in different ways. Just simply in development, they're called T cells because they develop in the thymus. They undergo a selection process where they have to learn. All of us have different HLA molecules. So in the thymus, they have to learn which HLA molecules you have so that they can see peptide fragments. And that's different in everybody. And so they undergo things called positive negative selection, i.e., they have to see self to survive. They can't see self so well as they do autoimmune disease. So there's a sweet spot that they have to function. And then in the peripheral responses, they have a huge level of responses they can do. So if you've heard of terms of cytokines, T cells make basically every cytokine from N-1 to the 30s. If you look at it. They have different functions. Some are called Tregs, which are regulatory T cells. There's a effector cell, an H. If you ever see something that's TH, it's an effector cell of some sort. It's a helper cell. It's providing help to macrophages and cells. There's memory cells. There's short-term effector cells, memory precursors. And about half my lab is autoimmune disease at the moment. That's because the number one risk factor for almost all autoimmune diseases is very good. Right? So that function of that is to present peptides, obviously T cells.
So that's the clarification. So how is peripheral response?
How does peripheral? Anytime you can detect T cells. So T cells that are circulating the body are usually in age status. So they can't make cytokines, they can't kill something. So they have to undergo a activation. And then they acquire cytokine kill, depending on. And then it's a system that's really got a lot of unusual features for it to function. It make it, if you think about it, really difficult to do. So the T cell, on the one hand, they're highly specific. They can detect single amino acid differences. So, for example, a viral escape mutant or a tumor neoantigen. But they're also, so they're highly specific. They can distinguish flu, COVID, whatever, but they're also cross-reactive. So there's not enough T cells in your body to see everything, but you can. So you have to have receptors that will see cross-reactivity. And in a case in point, with really about a decade ago, kind of changed people's thought. But if you take peripheral blood out of a human and look at the memory population, you will find memory T cells that should have been activated in something and had never seen that antigen. And the control was HIV. They were screening blood for HIV. And in people that aren't HIV infected, you can find all the hallmarks of the memory T cell that's seen HIV. So the only way that exists is if it cross-reacted with something else. So that T cell got activated. And as you age, as we're young or naive, as we age, we keep accumulating memory cells that can see antigens. Some of them you may not have ever seen. So you have this specificity, i.e., you want your T cell to just on the flu or a cold virus, but not just running around responding to tissues. You have that specificity, but it also has to cross-react. Also, they're extremely sensitive. So in a lab setting, they can see a single antigen on the surface of an APC is enough to see. Normal antigen presenting cells have 10 to the 5th molecules. So they can see one in this huge C, so they're very sensitive. Yet in general, they're also thought to be relatively low affinity. And so you've got specificity, cross-reactivity, sensitivity. And so these all these features don't make any sense with receptors. So how do you design a receptor system? And that's the question people have been asking for decades since receptors exist, and we're still trying to figure it out. And one reason you want to know this is it should allow you to engineer TCR or understand how to manipulate T cells. And so the obvious choice, the one I picked here to talk about, just because if you look at it, is tumor antigens. So cancer tumors are cells. So in general, you do not have high affinity T cells to tumor antigens. One reason it's difficult to get rid of them. And so since you can identify T cells that see any tumor in any individual, you can get those receptors, but they're low affinity. So people have this idea, let's re-engineer the TCRs and let's see if we can make them high affinity now and get rid of the tumor. And they had great success, or preliminary success. They could really do that. And so one of the early antigens was MAGE, a melanoma antigen, or tumors. And the preclinical testing was extremely promising. And then they put it in a patient. And so this is a famous one people talk about. They put it in a patient. 4 days later, the first patient, they transfused the T cells with this high affinity TCR that should be seen MAGE. 4 days later, it died. The immune system had attacked the heart. And so they looked at it and they analyzed it. They said they thought it was just, not just a random chance. So they put it in the second patient. The second patient lived 5 days and died. And so this time they did a much closer look, and they had T cells like crazy. And so then that led to this study here, which they had tested the TCR against all kinds of tissues. But what they hadn't picked up was this cross-reactivity such that they increased the affinity for the MAGE antigen to get rid of the tumor. But they also now created a new cross-reactivity with cell. Because remember, we always have to distinguish cell from us. MAGE antigen has been looked at the same time. Another group out of the NIH, they stuck it in going after tumors, and they were their TCR attacked the nervous system. And they don't know exactly what it is, but they seem to think it's cross-reactive MAGE. They put it in 9 patients. They also killed 2 people. And so, and a third one had got Parkinson's, so the immune system attacked, broke tolerance. And even if you do cancer therapies right now, the immunotherapies that are used in general are breaking tolerance. So a lot of people who get immunotherapies and cancer right now get secondary autoimmune disease because you're taking the control of autoimmune disease from the immune system. And you get diabetes, arthritis. And the last slide, I hopefully we'll get back to this particular TCR that attacked the heart. That's in to look at, if you want to look at the immune response, you really want to know how many cells are responding. So you look at frequency. So you start from very rare cells to any antigen, and they expand. So you're looking at frequencies. There's ways to do that. Probably the most popular way is something called peptide MHC tetramer, which is the diagram here. The affinity. So basically, what this is MHC with a peptide, and a single one of the molecules is too low of affinity to be detected. So what someone had a brilliant idea was to take advantage of the biotin streptavidin system, use streptavidin and biotin and put 4 of them together. That creates something called increased avidity. So now you have 4 molecules in proximity that combine. That now does allow you to use flow cytometry to identify antigen T cells. I think it's one of the more fundamental advancements that can happen in the immune system in 20 years. And so now you can take samples from people, mice, or whatever you're looking at. You know the antigen. You can say you have 20% of your T cells see COVID, 10% of them see flu. So we can now actually quantify. Part of that system has to do with affinity on lifetime, and we'll talk about later. So CAR T cells is a good question. So what happened with the CAR T cells? So I kind of alluded to T cell receptors are thought about for being low affinity, and that was defined in solution when I showed you 3D measurements. In 2D, they're actually really. And so what the idea about a CAR T cell, what they did was they took antibodies, which are high affinity, and now mated them with the internal signaling path of a T cell. So you have an antibody specificity that's high affinity in solution, and put it on a T cell, now redirected. But it's actually a very interesting idea because antibody binding kinetics, and the lab's doing something on that, is very different from the T cell receptor. I already talked about affinity. So all the defects right now in CARs probably relate to the idea that T cell is used to seeing a T cell receptor trigger, which has certain binding kinetics. The antibody is very different. So a lot of CARs don't have very good lifespan, they don't have memory. So they're very good effectors. They'll give you this huge hit, but they kind of kill themselves, peter out. Many of them. Is it a trade-off? Yeah, so I think we'll get to that, but yeah, that, I mean, that would be part of the trade-off. In essence, that would have been the interpretation of the cross-reactivity issue. So they really jacked up the affinity of the TCR, not thinking, well, now I've changed reactivity everywhere. So normally that T cell receptor probably sees part antigen at a very, very low level, never triggered. But now you raise its affinity and it's going, oh yeah, let's trigger. It doesn't know self or non-self. It just knows what's the kinetics. And so then, if you go to an immunology seminar, that's why I just threw this in. This is just a mouse that's been given virus LCMV. It's called. It's probably given us more advancements in immunology than any other immune system. And if you take a tetramer to a peptide in that LCMV, and CD4 is, so this is a site geometry diagram about half of immunology. So you can do antibodies to label rest molecule CD4. Subsets T cells. If you do tetramer by CD, see these T cells see this particular image. You can just say how many T cells see that given image. So that's the, now you can validate vaccines, you can validate immune responses. That's what they would be doing. Yeah, the antigen's the same, but you will induce a polyclonal response. And so precursor frequency in general, precursor frequency to any given antigen is probably into the 10 to 100 total cells in the body. And so then you have the clonal sense, and then you have multiple cells that can respond. So yeah, you will have a polyclonal response. And depending on the response, there may be some clonal expansion. So it'll select some. So about half the lab does autoimmune disease. We do model system that is for MS. We use it by using an antigen called MOD, which is a myelin protein. You actually induce an immune response in the mouse to make it paralyzed. You put it in an environment and give it toxin, give it to the mouse. Several days later, start having progressive paralysis. This data is more data. The first MD PhD student, he made a tetramer that we could see log, so that was valuable system. They have a scoring system and no symptoms to death. Most mice in the system, the sick mouse in the system, they get a progressive. The tail gets cursed. When you do that, and he made this tetramer, this is the score over time. So we tracked our mice. The mice got sick about 20 days. They got peak paralysis, and about 2 stayed sick for 80 to 90 days because their CNS is messed up. We tracked using the tetramer that Joe made. We tracked it. Here's the global cytometry diagram. So you can see we saw 9% of the T cells see that antigen, and we could track over time how many T cells were in the CNS. That was a great advancement. Except we left us with this question: what are all the other T cells seeing in the CNS? So T cells, immune system cells don't go to the CNS and stay there unless they have a purpose. It's a pretty privileged site. You don't want active immune responses going on there because you can have secondary. So that we had to figure out why would that happen. And so part of that, then again using cytometry, we can. If you look in this diagram here, in this animal system here, we had 5% of our tetramer positive or antigen responsive. Then we also track 2 different cytokines. So those are effector functions that T cells make. Interferon gamma and IL-17, it could be anything. And for both of those, you had 10% of the T cells responded. So more cells responded to antigen than the tetramer released. So again, what's going on here? Another way to look at antigen reactivity is you can have reporter mice. So you can make a reporter mouse that sees interferon gamma that is also GFP positive. So now I can ask the question, how many cells made interferon gamma? And so when we looked at the reporter mouse in our EAE, 50% of them are antigen reactive. And so now you're asking the question, if you look at this, well, how many antigen reactive cells in the CNS? Because remember I told you frequency is important. How do you tell them for the immune response? And so we have single digits of tetramer positive by intracellular cytokine staining, looking at the proteins in the cell. about 10% or 20% together. 40% or more were making cytokines. So how many antigen-reactive cells are there, and what's going on? And so, which is the correct number. So then we started thinking about what's going on with the tetramer, right? And so one thing you can think about here is, I mentioned a term called avidity. It's a term used in the immune system, especially for antibodies. So remember, an antibody has at least 2 arms to see the same antigen. That's why they're so good at seeing antigens, because you bind with this one, and right next to it, you have a second one, so it changes the binding kinetics. So it binds better. And so that's what increases avidity, and that's what this tetramer does for T cells. So if you have an affinity interaction, if you're just looking at reactions like a chemist, take reaction A plus B, you get that complex. Only difficulty here is we have proteins, right? So I have my TCR protein, we have my MHC protein, and then I have TCR MHC. And then I always like to point out the crystallography. This is an MHC molecule. And if you look at it carefully, they've turned it so it looks like a moose. So this is the nose of the moose, and then here's his antlers, and then the peptide sits between the antlers and top. And I mentioned to you that normally, if you want to look at affinity, you do it in solution, right? So that you put a molecule. This is an SPR, surface plasmon resonance system. You put one protein on a surface, and then you flow your second protein over in solution, and you can get binding kinetics. And basically, this is an example curve, but you need to use purified proteins. It's a solution phase binding event, so it's in 3 dimensions because the protein can rotate in every dimension. The affinity measurements are in a molarity range, right? So for TCR, they're micromolar ranges. And based upon that, that's where I said people think that T cell receptors are. They're barely at the detectable level. Problem with this was when everyone does this data in the early 2000s, it didn't explain T cell biology. That's why I was open to this idea of talking to bioengineers about another way to explain kinetics. How do proteins work? And the reason for that is this is really what the surface looks like. So again, here's our MHC and TCR and our peptide. But these things are sitting between membranes, and the TCR is connected to the actin side of the skeleton. It's got a second protein called a co-receptor that comes help it. It's got a whole series of proteins called CD3 that are its signaling components. So it's a much more complex system than what we were looking at. And especially when you look at this 2-dimensional, it's locked in place, right? It can only go in 2 dimensions. What really blew my mind was affinity in 2D is an area, right? Because it can only move in 2D. So it's not molarity anymore. We're actually talking about meters squared. And what really got us excited was it had much more increased sensitivity. So it could actually measure T cells, TCRs, and it could interrogate a large number of cells. I can do this to any immune response called immune response. And it also seemed to explain T cell biology. So that was, that ended up to be a Nature paper in 2010, because the 2D measurements then did explain biology a lot better. And the interesting thing is that when people think about it, 2D and 3D are not translatable. So no one's been able to take any protein, to take protein interaction, take a 3D measurement in the solution and get the 2D. So this is how we do it. So in 2D, what we have is a microscope, and we have micropipettes. And when we do the pipettes, we use suction to put a T cell on one pipette, and our sensor is a red blood cell. And the reason we use a red blood cell is they're very flexible. So their membrane will flex with 1 piconewton force, so they're easy to flex with the capillaries, and you can easily get a lot of them, and you can functionalize them any way you want. You can put any protein you want. So any receptor ligand interaction can be looked at in the system. So what I said here is we have a T cell on one side, a red blood cell on the other, and what we do is we touch them together, hold them together for a period of time, 2 seconds, and then we pull them apart. And if the antigen ligand interacted, the red blood cell membrane will stretch, and we just count that, or it doesn't stretch. It's a binary system, it either binds or it doesn't bind. And so this is a movie that the students and postdocs have to do. The first time it won't bind. The second time, the first time it didn't bind. The second time you might see the red blood cell membrane flex. If you caught that, that's what the postdocs and students are doing, and we count 50 times for each T cell, so then we get an affinity measurement for each T cell that way. So if anyone wants to do this, feel free. I'm looking for volunteers. But again, just diagrammatically, but the system is a T cell here with a red blood cell, and you put the peptide ABC on, and besides getting affinity, it gives frequency. So this is just showing us color-coded range of T cells that bound red blood cells, but some of them don't, right? 100% of your T cells can never see that antigen system. And then based upon a simple model, as long as we know the density of your receptor and ligand, you can solve for affinity. And so you solve for affinity, the ones that don't bind, you can solve for affinity, so they drop out of the system. That's how we get our affinity measurements. So now going back to our MS model or EA model, we have tetramer staining here, and then we did our micropipette assay. And what you see is we have a wide range of cells that binding a adhesion. This is just a control showing specificity. You don't always show the control, but we always run it. So these T cells are all specific for log antigen. And when we looked at the frequency, then remember it's tetramers I've shown you many times is in single digits. But by the micropipette now, we were in the 60 to 70% of the T cells in the CNS C antigen. So the reason the tetramers weren't very effective is they have a cutoff, right? So they only work in a certain affinity range, and every T cell below that affinity drop out. So they're good. They see antigen reactive cells. They dismiss all the low affinities. And depending on your immune response, you might have a lot of low affinities, such to cell, such to tumors. This is just a diagram showing that was a CD4 T cell. The other flavor of T cells are called CD8s. Just showing that we can see a wide range of affinity for every response. This is the LCMV virus CD8. This is two malaria antigens that you also give a wide response. This is a polyoma antigen. And so every T cell antigen, we look at a lot of different antigens. You know the antigen, and we get response of high and low affinity cells. But what happens is, remember, tetramers only see the highest ones. And so here what we did was, this is the more cells showing you the breadth of response for the island antigen. And here we generated two clonal populations. One's called V8, the other one's called 2D2. 2D2 is a T cell transgenic, i.e. you get all the T cells in the mouse are clonal for that antigen. It was made by the Harvard group. It's anyone who does EAE in the nation will have these T cells. V8, we made here. And I just compare it to the virus response here, just to give you an idea. This is an antiviral T cell on the high affinity range, but you see this wide range of affinity. And the title of this talk of this slide is "Affinity Does Not Explain Autoimmune Disease." And this T cell here, the low affinity, is actually below detection on my system. But everyone in the nation knows it generates disease. So even my system, which is very sensitive, it has a cutoff, affinity cutoff. And so low affinity does not actually dictate the T cell system whether you're going to get a response. That's actually quite interesting to think about. Here's just showing you that the main difference here, since affinity didn't explain it, low and high cells, they get sick just with different kinetics. So affinity determined kinetics, it didn't determine whether we would get a response. And so is affinity the only way to think about immune responses? And no, it's not really, because receptor ligand interactions, especially in biology, off rates or the lifetime of the interaction is often a determining factor. And so how well those proteins interact, and it can be called many different things. It can be called efficacy, potency, intrinsic activity. For example, you can have compounds in solution that have similar affinity, but their on-and-off rates might be different, and you'll get different effects. So that's why. So the question is, how do bond lifetimes vary in T cells? And so what we did here was I had the postdoc, instead of touching them together, which is what we do to drive affinity, just take the T cell and hold it up next to the red blood cell and see what would happen. And this movie's going to take 18 minutes. And if you look at the T cell over time, you see this T cell is getting really active, and the only proteins of interaction in the system are TCR and peptide. So antigen is enough to make a T cell do this. And eventually the T cell pulls so hard or kills the red blood cell that it pulls it out like that. And that was being healed by suction. So any T cell you do this to, if you hold it against something, it will wrap itself around. And the interesting thing is, a cell that's moving like this, that's generating forces. So on the surface, you are generating forces, and those forces are going to affect my life. And so that's what we got into the next measurements. And so we used we made, so the micropipette system is actually, the one I show you for affinity is actually fairly simple. In fact, all our original data is saved to VCR tapes. All these tapes to VCR. I still have a VCR player in my office that I can show anyone to, in case I have to pull it out. But yeah, it wasn't a database. So, but anyway, this one is a little more sensitive, a little far advancement, because to do force, so the affinity is a single cell measurement. For the force, we're going to do single molecule. So we're going to skew the system so we can do single molecule systems. We still have our red blood cell sensor, and we have a T cell. We've added a bead here, and we put the bead on the red blood cell. The red blood cell, remember, is flexible. Bead is what we put our ligand on, and what we're going to do is we're going to use a camera to contract the edge of the bead. And by doing that, we can now move the bead around, see if we get a bond lifetime, and measure the bond lifetime. And so what the data looks like here is Brownian motion. That's the edge of the bead here. That's just sitting there. The pipette is moved forward by the piezo. That pushes the bead into the membrane of the red cell. You hold it together for a period of time. Now you stretch it back beyond the starting point. And if you get a bond formation, the bead will go all the way. And now you can get a bond lifetime. And then eventually that bond lifetime breaks and the bead goes back to static. And so that's what this system does. And so this is just using my cell phone to take a picture over the, hopefully you can see this over the shoulder of one of my MD PhD students. We have a red blood cell and a T cell, and you can see the bead very clearly here. And this is the second screen is showing the previous run. So here you can see the where the bead was. It was pushed in. And when we pulled back here, there was no bond formation because the bead stayed. And to make sure this is single molecule, we set the system up, the density, such that we get binding events less than 10%. So we're basically doing Poisson distribution to get single bonding events. We can see sometimes two binding events because you'll see two bond lifetimes, and it'll be a bond lifetime and it'll break and get a second lifetime. But that's what we measure. And in doing that, normally you would think about when you pull on a bond that it would break faster, right? If you're going to apply force to something, you should break. That's not what we saw for the T cell receptor. And in fact, what we saw was as you applied force, the lifetime got longer. And so we did this, and remember I mentioned to you that T cells can see single amino substitutions. We compared it to 3 different peptide antigens that vary by 1 amino acid, and they activate this T cell differentially. They're widely used. It's probably one of the most widely used systems. And what we did was, is you look at the different amino acids going with their affinity and lifetimes, the lifetime changed, and also what changed is where the peak lifetime is. So we got this thing called a catch bond. And as you pulled on it, you got a bigger bond lifetimes. And as people, these are all papers that I've been involved with. The peak bond lifetime for T cells in general is at 10 picomoles. That's also been confirmed by other groups. And so this is a series of papers. Again, just remember. And really, when we think about catch bonds, we think about what came to us one day was one of the vendors in our plasticware. They gave us a Chinese finger trap flavor, right? One of the paper ones. And all of a sudden, I realized that's probably one way to think of this. You know, if you stick your finger in a Chinese finger trap and pull on it, it binds better. And so that's what happens to the T cell receptor. And eventually, you get to a point in a Chinese fingerprint that you're either going to break your fingers or the trap, but you pull it apart, and that's exactly what happens with the T cell. And so what that does is, this is just a summary of catch bonds. So they were originally proposed a long time ago. No one had the technology to find it. Finally, in 2003, using the system I used, selectins were the first subset that showed the catch bond. LFA1 ICAMs are the integrins. That's probably the one, if anyone knows about catch bond biology or proteins that bind better under force, it's probably due to the integrins, because LFAs or similar integrins are very widely known. Then our work, it was a Cell paper in 2014 that showed the TCR catch bonds. I also included NASH papers. So some people with you. B cell receptors also do catch bonds. Less looked at whatever reason, in T cells. And some of the original work came from bacterial adhesions. They also bind and do, which would make sense, right? So if you had a protein, you want to stop a bacteria on a cell and it's in flow, it would make sense that as the flow is pulling, that it would bind better, right? So that's what a catch bond does. But T cells are probably doing it for another reason. In fact, what reason I'm thinking of, what we propose, is it's to dictate and decide quality of that interaction, right? So the bond lifetime now gives you a huge range of abilities for different effector functions. So remember, all those effector functions I talked about, they could be controlled by bond lifetime. And so that's what we're thinking of. If I go back to our autoimmune system, here is again our 2D2 and V8, our two transgenics, lower and high affinity. We're looking at their bond lifetimes. And what we discovered is when we looked in the CNS, these cells, when they're naive, i.e. they weren't activated, their bond lifetimes are relatively short. But when they go cause disease, they now have long bond lifetime, and in fact, look as good as the viral response. And if you look at the viral response, naive and activated T cell doesn't change. So this to us was the dark side of an autoimmune T cell response. So Tumors changed the bond lifetime and made these T cells dangerous. And so right now we're trying to figure out how that happened, why it happened, the mechanism of what it happened. As I said, we see any immune response with an antigen, that optimal response will do this catch bond and probably peaks at 10 picometers. And bond lifetime probably was dictating the amount of whatever the effector functions are. So we look at catch bonds, and then autoimmune T cells differ from ones in the periphery. So basically it comes up to this idea that affinity is not enough. So most people think between receptor and ligands, affinity is what's going to dictate response. For T cell receptors, that is not true. I showed you low affinity T cells in autoimmune disease give you disease. That's well known. And one of the reasons I started thinking about quality. So if you've had an immunology class, you probably know that there's a genetic mechanism to get a huge repertoire of T cell receptors, right? So a huge number. Predicted numbers for T cell receptor could be at least 10 to the 18th. That's a large number. In fact, that's more. That number is bigger than the number of cells in your body, right? So this is a huge number. And so if you have all that genetic variation, how does that receptor always bind in the same way to peptide MHC? There's no reason to believe that you might not have other binding orientations, right? This is genetics that drive TCR generation is random. And so we started looking at, and then there was this study out of the NIH by Singer's lab. He took a mouse that had no MHC. So you would think there should be no T cells, but they got T cells. And what those receptors saw, they saw other proteins. So at some level, there are receptors that come out that aren't going to see antigen in the right way. So this is quality. So in 2016, a group out of Australia, I hope the resolution is good enough, identified flu-reactive T cell receptors that didn't trigger. They were high affinity because they were tetramer positive, but they didn't activate T cells and wanted to know why. They crystallized them. And if you look at the orientation here, the way they drew the orientation, the signaling proteins are here, and these ones were 180 degrees backwards. So they reversed. They called them reversal TCRs. And they postulated since they were backwards, that's why they didn't move. So as soon as we saw that data, we had set the question, what would they do with a catch bond? And so that was this data. This is the receptors here. This is the affinity measurements. We actually have 4 of them. We did the affinity measurements. They're all high affinity, i.e. you can measure the affinity. And these are the 2 that were in the normal. This is the TCR, a single TCR in a normal orientation. It forms a nice catch bond. And the ones that reversed don't form a catch bond at all. And what this is called is a slip bond, i.e. as you apply force, it breaks right away. And so the reason, the definition of why these receptors that are reversed don't work is they can't form a catch bond. So again, it proves catch bond is required for T cell activation. So that was published 2 years ago. This is another one. I was working with a biochemist at Notre Dame. He has this is a receptor he got out of a library for human TCRs. And he said it binds every HLA, every MHC molecule has. So he thought the crystal would be interesting. And this thing is really weird. It binds to the side of the MHC molecule. this is the peptide up here, so that's where TCR would normally bind. So it binds, it has affinity because he can crystallize it, so it definitely has affinity. We did the force on it, and it's a slip-on. So again, it's quality control. It's a receptor that doesn't bind in the right orientation, and it can't do the bond lifetime necessary to activate this. So perfectly made perfect sense. Another example is came out of a collaborations with Chris Garcia at Stanford University. He identified 2 T-cell receptors that see HIV peptides. So he identified these receptors out of a receptor library, peptide library. And here is the tetramer staining. You can't really see the overlap. So they're the same affinity, more or less, by tetramer, so they bind to that. Yet this one here activates cells and makes cytokine, in this case, called interleukin-2. The other one doesn't. So he asked the question, he could not figure out why they didn't activate cells. He has the crystal structures, they look identical. He had no reason to. I said, we'll look at it. My guess is if we slip, this is catch. This is just our affinity measurement, both are high affinity. And the one that activates, the blue one, gives us a catch bond. The one that doesn't activate is a slip bond. So what was interesting about this and why this got into Cell was that this is that slip on here. We could take the TCR now and make amino acid substitutions and now engineer in catch bonds. And so now these gave you 3 different response levels. We actually did many more than that, but this is just showing 3 bond lifetimes. They vary on how well they trigger the cell based upon the bond lifetime under. And so, that was an exciting finding. But now I wanted to get back to, and again, this just summarizes. We believe the lifetime effect of the catch bond, how long you have, matters. And so a lower bond lifetime is going to give you a different effector profile than shorter bond lifetime, different than longer. And this is just a diagram showing the TCR, peptide MHC, co-receptor, and the idea that longer bond lifetime effectors. So going back to the original one where I told you there was cross-reactivity to MAGE and we killed people because they attacked the heart. So the end of this recent paper in Science was we took that TCR and looked at it again and saying, what would it do? And this is the original parent TCR. Remember I told you it came out of a patient, but it didn't get rid of the tumor. So there was something that's not great about it. So it gives you weak bond lifetimes. These are very lifetimes here, 2.4 seconds. This is the engineered lifetimes that they put into patients. And you can see now they had a great bond lifetime. And so that was problematic. And so then what we looked at in this study was, could we engineer it differently? Looking at bond lifetimes, could we identify a receptor that still had great reactivity, i.e. catch bond, but not increased affinity? So that's all that science paper does. We screen for, try to maintain affinity to get rid of cross-reactivity, but increase the bond lifetime such that now we had effector functions, and predict that would get rid of tumors. And that's basically what we did. So the whole title of that paper is like engineering TCR reactivity. So that's where my lab sits right now. We do, we're looking at any T-cell antigen. I look for biology that gives me different responses. And so trying to figure out what is the definition of catch bond, what can we do mechanistically? We can do, we play with tumor TCRs, autoimmune TCRs, infection TCRs, different subsets of TCRs to see what effector function we get out of response, just based upon what the bond lifetime is, what kind of catch. So the summary is basically that optimal activation requires force for T cells. So remember back to that movie, T cells are always moving around. They're always touching and looking at MHCs. They're applying force to all the proteins. I only showed you a handful of proteins that have catch bonds. It grows all the time. CD44 is a catch bond. Other surface proteins that you think between lymphocytes or cells in cell interactions, since it's in 2D, and if they're moving and you can apply forces, I'm guessing the bond lifetime is controlled by this. It requires a cytoskeleton for the most active format data I didn't show you. And it will allow us to have a wide range of outcomes. And I think it's the quality control system. I think it's how T cells can distinguish self versus non-self, how you can survive, or give you full effector functions, and even distinguish between different effectors. And so again, one of the common questions in immunology is, do you have enough range of sensitivity to see all diverse antigens? And I think if you think about what I tried to explain to you in genetics, there is. It's not just an affinity system. We have multiple kinds of bonds, catch versus slip. We have a wide affinity range. So remember, I showed you antigens at least 10,000 normally. Lifetime also has a range of, our shortest are in the 1 seconds, 0.1 seconds to 10 seconds. The amount of force also varies somewhat, but for most responses to date, it's around 10 ppm. You could, in theory, think about the slope of it having effects. Other proteins having effects. Also, T cells have the ability to count. I told you a single antigen is enough to trigger the cell, but you could anticipate an antigen presenting some having 100 antigens would be very different. And the T cell response to these 100 antigens, you trigger 100 times at once versus we haven't looked at that. So I think there's plenty of diversity for to really run the T cell response. And then the final, when we think about affinity dictates this interaction here, you get a TCR binds MHC, you have to have affinity for the system to work, right? Once that affinity occurs, that starts a signaling event. That affinity comes in and feeds into the system, and then you get a feedback via the actin cytoskeleton that starts applying more force. So I think the way I see the system is you activate the system and the immune system, the actin cytoskeleton keeps applying force till it breaks. So it tests every bond until it finds its breaking point, and that tells you what kind of response. And then that also will then feedback. So just end up with acknowledgements then. This is a lot of my lab, a lot of the work. Well, she came with me from memory. She was a postdoc there, and now we're a faculty member here. And Bayu was the first author. He's got a PhD in biomedical engineering from WashU, and he was at Georgia Tech, and that's where we learned all of our molecules. And then about 6 weeks before I moved to Utah, I said I wanted to come here and learn immunology. And so he's had a very steep curve. I can't believe it just changed. It took us a long time to, as you probably would realize, for the engineers system on understanding forces and all that, and us trying to teach them T cells. We throw a lot of radiations at people, and it's a complex system, so it's a steep learning. And then Bayu and Mo have been doing it the longest. Mo's probably done the most different T cell antigens. Looking at it, and then we have a lot of collaborations right now. So Tracy has showed some data. Matt Williams. So I have a grant with Tracy on plasmodium antigens. Probably the escape mechanism really focuses on immune response. Probably why there isn't a very good vaccine. Matt Williams, we just got my fall has been totally amazing. That beyond my wildest dreams, we heard on multiple grants. And we put grants in all the time. I put a lot of grants. And we had some in the study section, which never happened. I'm really mad at the NIH. Had 2 in the same study section. And that's never right. And then the scores came in, and both of them were well under the funding ratios. So that was a great thing. I told the lab that, and I knew that this was the only got a little concerned there, but then I reminded them that. And then we heard on Maria, I work with type 1 diabetes, so a grant with her got also. And then Bayu actually got an R1 too. Just got Paul has been beyond me. Brian Baker, I do crystallography with, but also modeling. Yeah, John Altman, the NIH. I told you the tetramers are really what move immune systems forward. So they made money out for a tetramer course. So if you know the antigen, you can get the NIH will give you free samples for free monomer made. So I don't even have to make the antigens I do. Only downside of that is they're not happy with me because I guess I'm the biggest user in the nation on monomers. In fact, they tell me that they meet monthly every other week to decide who's getting scared at some point they're going to cut me off because I don't know what to do. I don't go have to make all these proteins ourselves. If you look at Well, I'm really concerned. Every time we send something to the NIH where they reply back to me, I'm scared. I'm emails. I'm thinking they're going to cut me. Or they're going to say, enough is enough. So anyway, but that'll take any questions. Thank you so much.
That was the